send a copy to chat. Okay. Hi,
Okay.
everyone. Today, welcome to the Spatial Omics Seminar Series 7. And we are very excited to have Xiao Wang from Broad Institute of MIT and Harvard. And she's going to talk us about spatial transcriptional profiling in 3D and also functional relevance and functional analysis in tissues. Briefly, Xiao is currently a core institute member of the Broad Institute of MIT and Harvard, an assistant professor in the Department of Chemistry at MIT. She started her lab in 2019 to develop and apply new chemical, biophysical, and genomic tools to better understand brain function and dysfunction at the molecular level. And previously, Wang was a postdoc at Stanford with Professor Carl Wiesero, where she was a fellow of the Life Science Research Foundation. And at Stanford, she developed comprehensive methods for analyzing RNA in intact tissues. And I think the method was called Starmap. And earlier, she also received her PhD from, I think, University of Chicago, then a BS degree in Peking University in China. And she also received various awards, including the DP2, NIH DP2, a New Innovative Award, and also a Science Scholar Award. And without then further ado, actually, I'll turn over to Dr. Wang for her exciting talk. And this is all yours, Xiao.
Thanks very much for the introduction. Hello, everyone. I'm really happy to be here to share with you my research in the past 2 years. So I think I just want to briefly explain why my title is different from the initial in the initial version. So I think initially I'm trying to talk about how we can translating spatial atlas to like tissue function, but then I realized we're still like multiple steps away from achieving that goal. So I'm stepping back a little bit, just talking about a really exciting development we have in the lab, how we can enable multimodal in situ analysis of single cell transcriptional states. So I think all these like single cell related cell states will finally eventually emerge into our understanding of tissue function. So we're just like taking baby steps to first enable tools to better analyze the biochemical contents inside the cell, and then next try to correlate single cell states both molecularly and functionally. And ultimately, we hope we can have a more comprehensive map about how single cells comes together to give rise to tissue function. Okay, so I think the story begins with just taking back to thinking about how we can best characterize the biological systems. So from one side, we can see all the biological like cells and tissues, they are ultimately determined by the DNA sequence. So sequencing is definitely one cornerstone technology enable us to decode the biological information. From the other side, I think microscope is another very, very important analytical tools that enable us to first discover the structure of a cell, and then it's still the most vital tools to study the physical form, and together with all these like fluorescent reporters, we can eventually understand the biological contents, including cell morphology and tissue morphology. But traditionally, those two types of measurements usually happens in two different samples because when we do sequencing, we always need to like purify DNA and RNA from the tissues. So it's actually there's a disconnection if we actually want to linking the genetic information with morphological information. But eventually just giving what happened in the cell starts from DNA transcription and all the way to protein translation and cellular function. It would be nice to actually doing sequencing and the microscope in the same sample so that we can maximize our capability to correlate the DNA sequence with like a phenotype with phenotype observations in order to better decode the gene to function relationships. But I think for people who are here today, it's already enthusiastic about all these spatial omics tools. So I don't want to spend more time to persuading you that the spatial omics is very important. But I just want to further introduce why it is particularly important in the study of the brain. Because the brain, it's a special organ, is not just a collection of single cells, it's a network of single cells. With sequencing, we typically lose the context of the network. If we're thinking about different fields of neuroscience, there are so many people just to try to mapping the connecting diagram of the brain structure, and also as well as try to study the brain online, which means they will conduct in vivo recording about and mapping of brain activity. So if we could have these spatial omics tools, that is to say, we can actually also gathering high-dimensional molecular information as a format of image, then we can have the opportunities to actually register molecular information with the structure information and further with activity information. So that will be the most comprehensive way that enable us to try to figure out how brain works. So through the rest of my study, I will try to make this comparison that thinking about the brain or any other organ in general as a PI, I think it's definitely a oversimplified comparison, but I just hope to using this guide my thoughts about why do we want to do this type of multimodal in situ analysis and how we what are the informations and the goals we try to achieve there. So yeah, as I just mentioned, single cell sequencing has totally revolutionized our understanding of cell types, right? So previously people defined cell types by morphology and by function, but the ultimately most accurate way to define a cell type is by molecule. Is by their biochemical contents and molecules, which is what are the genes expressed in a certain cell. And with the spatial omics, we further gathered the capability of restore those single cell molecular profiles in the tissue autonomy, which means we can see the tissue structure and with their single cell expression profiles. So that's kind of like where we got started. First, we want to create a really comprehensive single cell resolved spatial atlas so that we can better create this type of molecular defined structures. I think that's where we started. We aim to achieve spatially resolved readout, highly multiplexed readout of our expression inside the mouse brain so that we can go there to reconstruct this spatial atlas of molecular defined cell types. I think the first technology I developed is at Stanford with a bunch of stellar scientists at called the Swiss lab at bioengineering of Stanford. So the technology we developed is called Starmap. It basically contains 3 steps. First, we design really high specific probes so that we utilize a pair of probes that can specifically recognize the specific RNA molecules inside the tissues. And because we have a pair of probes, only when they are both hybridized correctly in the same on the target RNA, the signal can be amplified. So it's kind of like already a selectivity design that we can enable specific amplification of the signals because a single hybridization probes will always problematic and can have a lot of background. And after that, because we have this rolling circle amplification in a very specific way, so we can further adding chemical modifications during this DNA amplification and cross-linking that newly generated cDNA amplicons with the hydrogel matrix. And then we can digest all the other unwanted molecules that may block the optical transparency. And with this hydrogel tissue chemistry, we can not only imaging thin tissue slides, we can also imaging really thick tissues to obtain a true 3D mapping of many organs. And lastly, lastly, we will have this like in situ sequencing as a readout, because we do encode a gene-specific barcodes in this like individual probes, and we designed the in situ sequencing in a way to have a two-base encoding scheme and also error correction mechanisms, so that this will ensure the best SNR and reduce the error rate in sequencing. So because of these three elements of design, we can have relatively high accurate readout for RNA expression inside single cells. So here is one example of like raw data from this like 2D Starmap measurements. So all these like individual dots you are seeing is individual RNA amplicons, and all these like small clusters of RNA amplicons is actually a cell. I will mention in the following slides how we can go from the RNA molecules into as a single cell like expression profiles. And we initially tried like very high resolution optical imaging with like a really small voxel size, like 100 nanometer, in X and Y dimensions, and 250 in along Z dimensions. This like oversampling will enable us to do deconvolution and to restore the most high resolution spatial information out of the tissue. And because, as I mentioned, we have tissue hydrogel embedding chemistry, we can totally do this in a relatively thick tissue slides. As I shown here, this is an example of a thick tissue slides with 100 micron thickness, which is corresponding roughly to 10 layers of cells in the mouse brain tissue. And this mapping can also be combined with other like fluorescent protein labeling to enable simultaneously mapping of cell types, and plus their like cell morphology, like the projection patterns of neurons. This is just like the first technology we developed in order to enable 2D and 3D RNA expression mapping in the mouse brain tissue. We first aim to further scaling up this technology, because what you are seeing here is actually just a very tiny part of the mouse brain. But if you, as many of you already know, the ultimate goal is we want to do even whole brain mouse brain mapping like at a scale in the future, so that we want to actually be able to scaling up this technology. I don't want to go to any technical details, but now we can actually mapping this like a whole brain mouse brain slides at a scale, and we also can do this in a relatively large piece of tissue, such as the brain of the mammoths and even human brain. So after that, we actually, after we scaling up the capability of data acquisition, we actually encounter another big challenge, is how we are going to analyze this data at a scale with without any manual or human bias. So I think firstly, what do we want to obtain? What do we want to achieve through this type of measurements? As I mentioned, if we're thinking about the way we define molecular cell types in with the single cell sequencing, it's actually we use dimensionality reduction a lot, so that we have a gene by cell matrix by more than 10,000 genes, but ultimately we may just get a dozen of different cell types. So we want to do the same thing for our spatial sequencing data. However, if we're thinking about the dimensionality reduction in space, it's actually more complicated, and there will also evolve how do we deal with the information of XYZ. So the other thing is, although we can see the patterns, every dots of RNA inside the tissues, but how we can define tissue regions and other biological meaningful structures from the data. Traditionally, when we're looking at the biological organs, we define tissue regions by autonomy and prior knowledge of tissue function. But now, for the first time, we will able to gathering so much molecular information. Can we further utilize those molecular information to define tissue structures? And lastly, like how we can do this with given the computational resource to do data analysis at the scale. I didn't mention previously, but the fluorescent image data we generated is actually a huge amount of terabytes at the terabyte scale. So how we can do computation efficiently with all this like a large amount of data. So the way we try to solve this is by developing a bottom-up hierarchical clustering approach. So just to say, to explain it in more detail, it's like after we got the raw data, we register the RNA molecules through multiple rounds of imaging. We will see this like a small like a gathering or clusters of dots, which mostly representing single cells. So the very first task is how can we achieve high resolution or high accurate cell segmentation? Because the cell segmentation step is as important, it's very important. Just thinking about single cell sequencing, the critical first step is tissue dissociation. Whereas in the domain of spatial omics, if we could do cell segmentation more accurately, which means we can also do cell typing and differential gene expression analysis more accurately. So first, we aim to increase the accuracy of cell segmentation and with minimum human bias. And the second, we also want to utilize this like gene expression information and single cell expression profiles to further define these like tissue regions and to study cell-cell interactions. So I will start with a brief introduction about how we can do this cell segmentation. So the way we think about this question is that we have two basic facts. One is there are higher density of RNA inside the cells. And secondly, if there are two different cell types next to each other, very likely they will also have a contrast of gene expression in adjacent cells. So we just want to take that two information into our consideration so that we can both thinking about both measuring the local density of RNA as well as their neighborhood gene expression composition around the RNA, so that we can do spatial clustering to first identify individual cells. So this is like what we got. So initially there's just like a bunch of dots and the individual dots representing a single RNA molecules. And after this spatial clustering, we term this approach as a cluster map. We can first achieve this like cell typing. And what you are seeing here, individual colors are randomly assigned to different cells. As you can see, we can have really high resolution cell typing, and some of them even can outline the shape of neurons. And secondly, then we can further utilize this information to conduct cell typing. So in the middle, what you can see here are just different colors are representing different cell types. You can see we can reconstruct the layered structure of the cortical regions, cortical layers in the mouse brain tissues. And then next, by using this kind of like a local gene expression matrix, we can further define tissue regions. As you can see here on the right, on the right panel, that the tissue regions that are defined by molecular gene expression aligns really well with the prior knowledge of the cortical layers of the mouse brain tissue. So that is just like one example showing that we can use a fully automated computational pipeline to achieve the function of cell segmentation and further tissue segmentation. Such a type of pipeline is totally applicable to 3D sick tissue data as well. Here is an example. Again, it's like a mouse cortical structures that you can see we can further do very high accurate cell segmentation and secondly tissue segmentation in 3D. Because the majority of the spatial omics data are conducted in 2D, but it's actually been challenging to even do this data analysis in 3D. But the cluster map approach, because they only utilize the proximity of RNA molecules with each other, so it can actually totally applicable to 3D data. So because we have now a really scalable experimental approach and computational approach to do cell typing inside the mouse brain, we first generated a really large scale mouse brain atlas with very high resolution. Here is just like one panel showing that we can do this, we can conduct a whole mouse brain cell typing with more than 1,000 gene measured directly at a 200 nanometer pixel size at million cells scale. And we can also do this at other organs species as well. And showing here, this is one example of mouse placenta. And this is also applicable to like sick tissues, like 3D structures, like human heart organoids. This is like what is the demonstration we have done so far. As a small summary in this session is that we, in the past 2 years, we first we first developed this like Starmap tools and also this cluster map computational pipeline in order to do high resolution gene expression profile and cell typing at the scale. Then the next step is after we can do this at a scale with high accuracy, what are the most exciting scientific problems we can solve, or what are the meaning of the molecular cell types? Are they true? Because for other people who have done this like cell typing research, you will know that there are different, there are just like so many different resolutions of cell typing and clustering you can do. But the question is, do we know they are real cell types? And how do we verify those molecular defined cell types? So the way we think about this question is first, although molecular cell typing won't be perfect, however, that's the most universal or unbiased way that we can define cell types. But more importantly, on the next stage is how we can assign functional readout with those molecular readouts so that we can achieve more accurate definition of cell types. We can also do in order to achieve better understanding of how the genes are contributing to tissue function. So again, putting this general question in the context of neuroscience, if we try to link these molecular cell types with functional readout, there are a few dimensions to think about. First, what are the functional readout for the brain? You can thinking about there would be like multiple ways you can define a cell function. But normally, if we're thinking about the brain function, we will thinking about electrophysiology. That is to say, first, there will be spikes of membrane voltage change in the neuronal cells. And this will also have this like calcium signaling component happens together with this like active action potentials. So then our thought is like, okay, can we actually link those molecular cell typing directly with either calcium imaging or voltage recording? So we start with in vivo calcium recording because that's a like a well-acknowledged way to decode like a cellular function. And the second, it's relatively easier in implementation. So this we first try to say, okay, let's do in vivo recording first and then come back to do Starmap, and so that we can register those functional readout with molecular readout. And this is through collaboration with Dr. Emily Silverstrak and also like Dr. Carl Desrius' lab in order to achieve this like functional annotation of a spatial cell atlas. So the structure that we studied is called medial habenula. It's a relatively small brain structure along the middle line of the mouse brain. However, it has a striking heterogeneity both in gene expression and in their behavior phenotypes. So as shown here, you can see already from the traditionally mapped in situ hybridization data, there's already like a large diversity of gene expression in this small area. And secondly, if you destroy different part of this brain structure, you actually see very different phenotypes. That is to say this brain structure is very important to many different behaviors. Then our question is, do we actually know those molecular cell types, or are expression patterns align with the functional organization of the brain circuits? So in order to do so, we first did these Starmap experiments with this like a small brain structure, medial habenula. From there, we found the three major types of neurons. I think these like a dorsal and ventral cell types are relatively well known. However, there's like a new cell types of this like lateral habenula neuronal type marked by TH genes. And after that, Emily constructed this transgenic line to enable calcium imaging of these like three different neuronal population. And she also conducted this like a behavior assay where the mice are trained to respond to a visual cue, which is light. If the mice see the light and go there to push the red button, and it can come back to receive water as rewarding. So basically, it's like a visual cued rewarding assay. As shown here, I think these two very well-known neurons, which is the TAC1 and the CHAT1 neurons, they are like responding to this like reward consumption of water. However, this like newly discovered neuronal types are actually responding to the visual cues when the light is turned on. So it's like basically predictive cues. So this is maybe a very specific example about how the functional readout, how the functions can be also segregated for different molecular cell types. And this is also a very strong indication that the molecular cell types we are measuring from the mouse brain will be extremely helpful for us to better understand the brain circuits in the context of cell types. And after these encouraging results, we are also thinking about how can we do this in a more accurate way. So ultimately, the most accurate readout of electrophysiology is still the change of electrical signals and voltages. So then our goal is, can we also do this, combine this like spatial atlas measurements with electrophysiology? This is a much more challenging task, and we are still on the way of developing it. But we just did a very small like proof of concept experiments with another organ that biological systems that do have very interesting electrophysiology patterns, but easier to measure. So this is through the collaboration with Dr. Jalil's lab at Harvard. The system we choose to demo this concept of integrating electrophysiology with gene expression, it's a human organoids, heart organoids. So it's a system that we can because their lab have developed this platform which called the cyborg organoids. They can grow the electrodes together with the heart, iPS cell derived progenitor cell populations to allow the full maturation of the heart organoids, but recording the electrophysiology signal simultaneously. Then by utilizing this like a very high accurate and scalable way of in tissue recording, we can first obtain the electrophysiology measurements of the trace the maturation and the different maturation of heart organoids over time. And then we can also conduct in situ sequencing based measurements of molecular profiling so that we can simultaneously get gene expression information. And then again, the question is, we wonder, does like gene expression actually reflect this like a cell, this like a gradual cell states transition along the maturation of heart organoids from progenitor cells to cardiomyocytes? So it's again the overarching goal of like a better linking gene expression with function. So as shown here, again, the results are very encouraging. What we found is if we just looking at the maturation of electrophysiological signal, which are characterized as the shape of the electrophysiology. This can only of those like spikes. So those can only be measured accurately through electrical recording, not calcium imaging. So this allows us to do very high accurate characterization of the electrophysiology of heart cells. And simultaneously, we can do in situ sequencing mapping in a 3D heart organoids at the scale, so that we can have this like a very comprehensive gene expression profile. And through this, we do see a alignment at the single cell resolution between these two types of measurements. And more importantly, when we join these two types of measurements together, we can even have a more accurate definition of like cell types or cell states in this context, which means these like multimodal measurements can enable us to achieve a more accurate like a definition of cell states and cell types. And more importantly, because we have this direct correlation between gene expression and electrophysiology, we can further correlate exactly which gene may contribute to which electrophysiological features. Through this type of cross-model analysis, we can actually better register genes involved in different pathways with their corresponding electrophysiological features, such as different ion channels and different organization of the cytoskeleton that will undergo drastic change during the maturation of the heart organoids. Those like gene to function correlation will serve as a basis for many important biological discoveries to better understand a lot of different biological processes. In this case, it's the maturation of heart organoids, but it could be applicable to other biological systems. So this is just a proof of concept example. We call it in situ electrosequencing to allow us to integrate electrophysiology with gene expression. So yeah, I think those two examples are just trying to demonstrate our concepts that how we can do functional annotations to molecular defined cell types. And this is only enabled by the measurements of spatial omics. Lastly, I just want to mention another example. What if things go wrong in the tissues? Because I think in real clinical applications, a lot of disease diagnostics are achieved by, or pathological studies are achieved by tissue histopathology measurements. So we just wondering, since tissue histopathology is also obtained as a format of images, can we actually using our molecular cell atlas image to register with tissue histopathology image so that our this kind of like spatial omics approach will better enable us to understand human disease. We are extremely interested in one of the neurodegeneration disease, which is Alzheimer's disease. So this disease has been mostly this is a major cause of dementia in aged population, and it has been mostly defined by two characteristics. Disease hallmarks. One is the formation of amyloid plaques. The other is by this intracellular degeneration of cytoskeleton, which is neurofibrillatory tangles. Of course, there are many other disease-related measurement matrices that can together define the Alzheimer's disease, but those two has been two characteristic markers. And then how we can, and on the other side, just thinking about how single cell omics has revolutionized the way people study disease. There's another paper from Li Huicai and Manolis Collins lab at MIT. They did this single cell sequencing in human Alzheimer's disease samples. Of course, there are many other labs in that domain has contributed a lot of knowledge about how the disease or reflected and caused at the single cell resolution, can we identify disease-associated cell populations, cell types, and genes? So those will be also a very important basis for us to understand the disease and further develop potential treatment strategies. So with this single cell measurements, first we can actually identify a lot of disease-associated subcell populations, but we actually lose the correspondence about how these single-cell transcriptomic profiles are correlated with the A beta and tau tangles, these two major disease hallmarks. So then to enable such a correlation, I think maybe you already heard the talk in this seminar series. One easy thing to do is you can create a tissue sandwich where you can measure this spatial transcriptomics data at a relatively moderate resolution. But however, you can do a tissue histopathology staining at adjacent tissue slides so that you can do this cross slides registration and try to restore what may be the gene expression change that associated with these protein aggregations. So this is certainly a very smart design, but it's not ideal. So then what we try to do is can we actually measure everything in the same tissue slice with a high spatial resolution? So then we develop a new version of Starmap. It's called Starmap Plus, where we try to integrate the measurements of proteins together with this in situ sequencing of RNA expressions. In this particular case, we integrated the readout of antibody-based staining of intracellular tau tangles and also dye-based extracellular staining of these A beta aggregations. And this we can also simultaneously measure thousands of genes inside these tissue slides. So then after we gathering this type of high dimensional information, we of course first will do dimensionality reduction so that we can see all the cell types near the A beta plaques and also the cells that are actually containing tau tangles. Because this is a relatively high resolution imaging, we can further register the differentially expressed genes in different cell types or cell populations that are near or overlapping with this disease progression markers. So this is just one example where you can see these mouse hippocampus regions and the cortical regions. And this zoomed in regions just to show two examples about you can different colors representing different cell types. You can see these there are different cell types surrounding the A beta plaques. First is microglia cells. They are the first responder of this the A beta aggregates, and then there will be other cell types, including astrocytes and oligodendrocytes, even oligodendrocytes precursor cells further surrounding these loci that indication of a cascade of inflammatory response propagated from microglia to astrocytes and oligodendrocytes. From the other side, we can also identify these tau tangles. They are mostly aggregated in CA1 projection neurons, and then there are also associated change in the axon bundles with particular subtypes of oligodendrocytes. So I don't want to go to too much details. I have maybe a few minutes left, but I just want to show you that the powerfulness of this approach. For example, this is the microglia cells we just mentioned. You can see they are strictly associated with disease progression, and they are also responding to a lot of different inflammatory responses, such as they are surrounding the A beta plaques and also trigger particular immune response pathways. And meanwhile, they are also triggering and associating with the emergence of tau tangles. And in general, we try to build a much higher resolution and comprehensive maps about what happened in Alzheimer's disease. This is still, as I'm showing here, this is just a summary about this subtypes of these disease-associated cell populations, as well as gene markers. But in the future, we do want to apply this to real human tissues to see what we can discover there. So this is still an ongoing effort. Yeah, so just as a summary of today's contents, we first aiming to develop a more accurate way to mapping this spatial atlas, so that those will be laid down as a foundation to enable us to further achieve functional registration or annotation of cell types with tissue functions, and further we can also register molecular cell types and cell states with disease progression. So in the future, I think we relatively automated the computational pipeline to generate tissue atlas, but I think there are a lot of work to do in this domain to further generate computational methods that allow better analysis, for example, for disease samples, and then such kind of like multimodal measurements will first be we are aiming for is to do as much as we could to achieve simultaneous measurements about like RNA, protein functions and readout in the tissue. But ultimately there will be a limit about how much we can measure simultaneously in one sample. Then it's also very important, since there are like spatial omics data generating from so many different labs, we will also require a computational approach to integrate these different types of measurements in their spatial context. And then in general, these multimodal measurements will be greatly accelerate the discovery of gene to function. So yeah, so and this type of methodology is definitely not only applicable to brain tissue, it will be applicable for so many different biological studies. So I think the overarching goal is spatial omics, especially this like image-based in situ sequencing approach, will enable this integration of genomics with bioimages. And also enables integration with both image-based spatial omics and chip-based spatial omics. So yeah, I think it will be very exciting to see those integrations happen in the future. Okay. So thanks everyone. I just want to thank all my lab members, at least a few names from my lab that leading these studies I described here, but there are many more folks in my labs that actually contributing to the excellent like research here. And also I want to thank all these collaborators. We have this like cluster map and this like hard organoids in situ electrophysiology was developed in collaboration with Dr. Jia at Harvard. And then we also have been collaborating with Emily and Carl's lab in order to do this like calcium imaging and in vivo behavior. And this like Starmap Plus and Alzheimer disease study was done in collaboration with Morgan Sheng's lab at MIT, BCS, and Broad. And also we have collaborators from Genetech, which give us a lot of biological insights about what happened along the disease progression of AD. And I also want to thank all these generous funding agencies. And thanks everyone here today for my talk, and I'm happy to answer any questions.
Thank you, Xiao, for this fantastic talk, describing a journey that you have experienced. And if you have any questions to Xiao, please feel free to unmute or post on the chat. Yeah, Suyan, please go ahead.
All right, thank you, Xiao. Thank you, Ahmet, and great talk, Xiao. So my question is about the Starmap technique. Can it measure RNA inside of the nucleus? And if so, could you take advantage of the nascent RNA are more maybe in the nucleus before they're transported to perform RNA velocity analysis, especially on the your work in collaboration with Professor Jia Liu. I see that from one day to another the transcriptome change. Could you maybe generate RNA velocity arrows on those to show that maybe like each day there's a trend to move towards the nest maybe?
Yeah, I think that's a great suggestion. It's definitely doable. We are on our way of developing this strategy. So I think definitely, I think especially from what there's one paper from Xiao Zhang's lab, right? By also this like in situ RNA velocity definitely will be a very important way to ask infer the trajectory of a cell expression, the progression of the cell lineage. So yeah, I think we will definitely try it out. Yeah, that's a great suggestion.
Great. But from your data, actually, you do have these amplicons inside of the nucleus too, if I understand correctly, right? The chemical reaction is feasible inside of the nucleus.
Yes.
It's not just cool. All right, thank you.
Yeah, so I think yeah, that's great. So I think sorry, we don't have enough resolution for this raw data, but sure, we do have RNA inside the nucleus and in the cytosol. So yeah, it's totally feasible to do RNA velocity by using this like compartment information. Yeah.
Very nice. Thank you.
I think there is one question on the chat. Can you read it or should I help you, Xiao?
Oh, let me click the chart.
Yeah, otherwise I can also read.
Yeah, so I think the question is, what's the benefits of using transcripts for cell segmentation rather than staining the cells? That's a great question. I encountered this question many times. So I think yes, if there are many organs and cell cultures that can be easily segmented by like dye staining, but there are also organs that are very hard to do dye staining. So the reason we want to do using RNA transcripts, we want to have a universal applicable approach, regardless what the organs there comes from, regardless of like organ morphology, so that we can achieve a usually automated and biased way to do this mapping for a lot of different organs. So yeah, I think in our first publication of Starmap, we used the nasal staining to outline the cell shape of neurons. That has been working well for neurons, but not working well for cell cultures and other tissues. So there are also other dye like WGA works pretty well for some of the organs, but not all cell types. So we just found in general, those dye staining has a strong preference or bias towards different cell types. So we see good cell typing results with some of those, but we want to have something universally applicable to all cell types. Yeah, that's kind of like the motivation for using RNA to do this cell segmentation. Yeah.
Wonderful. I think we have two questions, one from Wubing.
Yeah, hi. Exciting talk, and I have a kind of technical question regarding the hierarchical clustering in detecting the cellular communities for the like tissue layers. I suspect the single cell gene expansion and the spatial information are both incorporated in the analysis, and I wonder actually how the spatial information are used in the hierarchical clustering. Besides, I wonder because in the brain tissue might have clear tissue boundary, which might be easy to detect the functional cellular communities. And how about the spatial data in cancer? Because in the human microenvironment, multiple different cancer types are mixed together. Is the method still work or not?
Yeah, I think that's a great question. So what we do this like a tissue region identification is we put each cell in the center of analysis, and then we draw a circle and counting and thinking about the gene expression of their nearest neighbors. And then if all these cell populations in this like a neighborhood gene expression are similar enough to each other, they will be classified as tissue regions. The nice thing about this approach is even we have discontinued tissue regions, they will be this can still be safe accurately identified. This doesn't allow any like continuous tissue region. So that is to say, in your question, if even you have like different cancer cells and immune cells or other like normal cells mixed together, as long as they have a relatively distinguishable gene expression by themselves, plus their neighborhood gene expression information, they can be still safely identified. As an example showing here, this is a mouse placenta, and in the middle, it's actually the cell typing results. On the right is the tissue region results. You can see there are like some tissue regions. You can see the orange color here. They are actually pretty mixed with other tissue regions, but we can still identify them. Regarding the cancer and other regional analysis, I think we haven't tried, but I think it could work as long as they are distinguishable gene markers for those regions.
Thanks, it's fantastic.
Wonderful.
So basically we yeah, so in summary, so we basically utilize the neighborhood gene expression, but indeed we didn't actually need the like physical XYZ in a larger scale. So yeah, it's kind of like a local scan of a neighborhood recognition. Yeah.
Wonderful. We have another question from his son.
Hi, thanks for the wonderful talk. So when you're talking about this distance from the plaque and things like that, you also have to think about the 3D as well, right? And as far as I understand, this technique is still optical, so probably go up to, I don't know, 100 micron thickness the most or so. How do you think that we can do the 3D profiling better? Because obviously distance is a 3D, the 3D characteristics.
Yeah, that's a great question. So I think for the examples we are showing here, this is still a piece of thin tissue slides, which means we mostly see one layer of cells within that thin tissue slides. So we did actually 2D spatial analysis for this like cell type, for this study, but in the future we do hope to do 3D analysis. If you I think I'll go back to still the first Starmap data. So we did this like a 3D like a local cell cluster analysis in mouse visual cortex. We do see 3D distance is much accurate than 2D distance. So I think next what we want to do is definitely to do this in a relatively thick tissue, so that we can do directly 3D measurements of distance. Yeah,
so when you say
agree, like 3D distance,
it's still 100 micron or so, right?
Oh no, for this thin tissue, it's just a 10 micron. So
right,
we
right.
cut
so
it.
If you want to go to thick tissue, I mean, you can't really go whatever that it allows, that the objective allows, right?
Yeah, definitely. So I think we normally do 100 micron to 200 micron measurements because we rely on very high resolution confocal imaging, yes, given the applicability of the objectives, the most comfortable range they work with will be somewhere between 100 to 300. We found beyond the 300, there will start to have this refractive index mismatching. So we rely on better microscopes like a light sheet in order to go even to thicker tissues. But yeah, routinely we do 100 to 200. For even thicker ones, we need a better microscope setting for that. Yeah.
Thank you.
Wonderful. I think we have now chat questions.
Yeah, so I think we have 2 more questions in the chat. Firstly, is about the transcript count. So are the transcript count are detected for each cell using? Oh yeah, so I think that's also a great question regarding how many transcripts we can detect per cell using this like 1000 gene panel. So I think it depends on what are the nature of that 1000 gene panel. So for example, if they are very highly expressed cell type markers, they are not necessarily expressed in all the cells, but in the right cell type, the expression could be very high. But there are also transcripts, they are relatively low expressed. So we may see like them has like a relatively low rate, like a few copies per cell. So I don't have a like a for sure answer to about like what's the dynamic range for our approach, but we have some for the Starmap and Starmap Plus, all the data released online. So if you want, we can take a look into the individual genes and the cell types that we mentioned here. So I think if we're thinking about the total transcript counts per cell, so and we typically observe somewhere between 200 to 1,000, and some in occasionally more than 1,000 like amplicons per cell. So that's why we kind of like mostly doing 1,000 gene measurements, including cell type markers and some activity related markers. And for this like Alzheimer's study, we scaled our panels to about 2,700 genes. We found it's also working pretty good. And we even try to do like even a lower we scaled back to like 64 genes, but really high like a dynamic range for individual genes. But what we found is in general, being able to measure more genes is more beneficial than measuring a few genes. So we kind of like, for example, if we just have measured 64 genes with individual gene wise, individual gene will have higher sensitivity, but that is actually not as beneficial as even we have like a compromise some of the sensitivity for individual genes, but we have a much larger gene panel that's more beneficial to identify the right gene expression pathways. Yeah, so hopefully I answered this question. Okay, the next question is how about neurons, RNA in the dendrites and axons. So indeed, the majority of the shape or the reads we recovered is actually from the cell body and maybe slightly in extension to the axons. However, if we want to track the axons over a long distance, it has been very challenging for reasons because all these like neurons, like dendrites and axons, they will have relatively similar gene expression, which means it's very hard for us to trace back which neuron, which amplicons or which RNA from which cell body over a long distance. So in order to make this happen, we may need to rely on, for example, like a rainbow type of labeling so that we can make individual neurons distinguishable from each other by some barcoding system, either RNA barcodes or protein barcodes. So as long as we can make individual neurons distinguishable from each other, we can then safely track the RNA back to the cell body. Otherwise, as I mentioned, if like adjacent neurons has relatively similar RNA expression profiles, we cannot trace it back to individual cell bodies over a long distance.
Wonderful.
Yeah.
And thank you. And if you have any questions, please feel free to post on the chat and or unmute yourself. But in the meantime, I can ask one question as we wait. So the correlation to the electrophysiology and transcription, it's amazing, right? You know, being able to see this. Could you comment on the time synchronization of this process? Like, what's the sensitivity of time, right? I mean, what kind of windows in neuroscience can we do this correlation for, right? And what do we do to extend this window, right? Is there any other kind of like innovation that we can think about?
Right, so I think just thinking about the time domains, for calcium imaging, I think it's relatively a shorter time window because there will be the problem of phototoxicity, right? So, and then there will also be issues like how we can do this in like over a really long period of time, like over months. But for the electrode-based recording, it's actually very reliable. especially I think there are like many new development inside the field that allow the tracing even the same neuron over months, right? So it's a very reliable tracing. So I think typically in situ sequencing is still a endpoint measurement. So whenever the measurements is done, we can do this. And it's just the time domain is more rely on how accurately in vivo measurements can do, right? So it's like, what's the temporal resolution? What are the time spans it can achieve? I think there are just a lot of exciting development in the domain of like soft electrode based in vivo recording that can like enable us to do this type of analysis.
Wonderful. Hopefully, yeah, we will have a live barcoding in the future. And we have two more questions, I think, in the chat as we waited.
Okay, so I think I see 3 questions. So first is comparing Starmap with Stereosig. Yeah, that's quite a great question. So I have been actually tracking the development of Stereosig. I think it's very exciting. So I think they achieved, according to paper, subcellularly resolved chip-based capturing of these spatial measurements. So I think in comparison with the Starmap and the Stereosig, there are maybe 3 major differences. First, it's Starmap is a targeted approach, which means we will design the probes and have a targeted gene list to start with. And the Stereosig is it's basically spatial capturing, so this naturally whole transcriptome capturing measurements. So regarding the sensitivity, I don't know. So I think maybe sometime we can do a side-by-side comparison. So that's the one thing about the gene measuring. And second, it's about the spatial dimensions. I think intrinsically, this image-based approach still has a highest spatial resolution, especially if we are, for example, we are caring about many subcellular events, even subcellular compartment like nuclear, cytosol, mitochondria, ER, etc. So I think this high spatial resolution is still very, at least important for us to better understand what happened in these like subcellular domains. And lastly, as I mentioned, it's like a spatial like Starmap because it's image-based, it's intrinsically a 3D measurement tools, whereas Stereosig, it's you can cut a few tissue slides, but for each of them, it's still a relatively 2D measurements. So yeah, that's another distinction. We do have this like 3D measurements capability. But I haven't done any side-by-side comparison by myself. Okay, so the second question is, what is magnification objective you use for imaging thicker tissue samples? That's a great question. So, you know, I think initially it's very hard to find such objective. We use 25x like a long working distance objective. It's like a water immersed one because that now allows the best matching between the refractive index of the immersion media versus the tissue slice, because it's basically hydrogel. So it has a refractive index similar to water. So we use 25x water immersed objective, long working distance, to imaging these like thick tissue samples. And I think especially given the development of 2-photon in vivo imaging and some of these like whole brain tissue clearing or whole organ tissue clearing technology, there are more and more such type of objective available. Like we're using like a Leica confocal, but it should be, there are many options for other microscopes as well. Okay, cool. So then there is also about whether it's Starmap can be applied to FFPE tissues. That's also a great question. We haven't tried somehow. So I think because you can see some successful measurements with other spatial omics, for example, this like a DIBT-seq is totally compatible with FFPE tissues. So I do think as long as the RNA is accessible for our probes, our approach should also be applicable to FFPE tissues, but I haven't tried it by myself yet. And for all these Starmap measurements, we also start with, fixed sieves like PFA or other fixed sieves. So it's basically also like, okay, that's actually another distinction with stereosig and other chip-based approaches. Like we typically work with fixed tissues, and for these like chip-based measurements, it typically starts with a fresh tissue. Yeah. I hope this answered your question. And okay, cool. So any other questions?
I think there's one more on the chat that I see, asking the detection efficiency of the current version of Starmap.
Oh yeah, so we did a side-by-side comparison actually in the first Starmap paper. I think according to our measurements, we compared with padlock-based, probes and other approaches. I think for the initial versions of in situ sequencing, it generally has much lower efficiency than SMFH. For example, there may be about 1%-ish efficiency in the earlier versions of in situ sequencing, because they all rely on reverse transcription, which greatly limited the efficiency because most of the time the enzyme didn't work well with the fixed tissues. So the reverse transcription has been a limitation of many of the in situ sequencing approaches. In our Starmap measurements, we didn't do reverse transcription. So our efficiency is somewhere between 10% and 50% of all the RNAs. So it's quite comparable with the single cell RNA sequencing. And we can actually achieve higher efficiency. The key point is how many probes you will design, right? So I think currently, if you design, for example, 4 or 5 probes per gene, you can have very high detection efficiency. But if there are occasions you want to measure more genes, for example, in the study of Alzheimer's disease, we actually only designed 2 probes per gene. So typically the detection efficiency is proportional to how many probes you design per gene. Yeah, so I think in general, the range of detection efficiency is around 10 to 50% of all the RNA population, depends on how many probes you design per gene. Yeah. So for SMFH, right, typically because they design like at least 20 probes per gene, that's why they have like higher efficiency, but it also requires like a lot of extra amounts of probes.
Correct, thank you. And I think we have covered most of the questions. And I think with this, let's thank to the speaker again, Xiao. Thank you. I'm gonna use this clapping sign. And I appreciated your talk and presenting to us the music, right? Journey of music and spatial omics. I really like these piano analogies actually. And before we also wrap up, I'd like to also announce that next week we will have another industry partner presenting from Miltani Biotech, and Amy Yablonsky is going to be presenting Level Up Immune Profiling of the Tumor Microenvironment, Deep Phenotyping and Target Discovery. And with this, we're going to be wrapping up today. And thank you, everyone. And thank you again, Xiao, for presenting this fantastic work. You're muted, sorry.
Oh yeah, thanks for the invitation, and I hope you have a great weekend. Okay.
Wonderful.
Bye.
And thank you, everyone. And I hope you guys all have a great weekend and hope to see you guys next week. Bye-bye for now. Thank you.